press the bell icon on the YouTube app and never miss another update.
We will continue the chapter Biotechnology Applications. We are continuing with the chapter Biotechnology Applications. In the last class, we were going through with biotechnology applications in the field of medicine. Under that, we have already discussed, especially there are around 30 recombinant therapeutics are in use worldwide, out of which the 12 are marketed in India. Under that heading, one of the recombinant therapeutic drugs, what exactly we discussed was the role of insulin. Well, as we all know that insulin is required to regulate or lower the glucose level in blood, and thereby the persons who are suffering from diabetes mellitus with higher glucose level, normally insulin is much required. We also require that insulin, being a protein hormone, it cannot be orally consumed because the protein hormone will undergo digestion and it results in amino acids. So thereby the insulin injections are given. As in the last class, as we discussed, the insulin will be same in other mammals also. Earlier, when recombinant therapy was not possible, technique was not possible, insulin extracted or isolated from slaughtered cows and pigs is to be given to diabetic patient, but it used to cause allergy to certain individuals. And also the amount of insulin from the slaughtered animals was very less. So due to this, the attempt was made to isolate a gene responsible for insulin from human pancreas cells and introduce those into the plasmids of E. coli, and culturing this transformed E. coli in bioreactor, one can get insulin. As we have already studied in the last class, functional insulin composed of two chains, one being alpha chain, the other being B chain. A chain with 21 amino acid and B chain with 30 amino acid to be joined by disulfide bonds to get that functional insulin. But in all mammals and even including humans, the insulin when produced will be produced in proinsulin form. The difference between the proinsulin and active insulin is, in proinsulin there will be another chain, that is C chain with 33 amino acids will be there, that will be removed in the functional insulin. And also, since this insulin is obtained from human genes through recombinant DNA technology, it was named as humulin. And it was in the year 1983, a US company by name Eli Lilly first produced and marketed this human insulin for the first time. That was exactly we had discussed in the earlier class. And the next concept was regarding molecular diagnosis. Molecular diagnosis. Molecular diagnosis. This is the point which we were started discussing in the last class. You know that proper therapy means treatment of a disease is possible through early diagnosis. Early diagnosis. If I tell diagnosis, you know that it is finding out what is the cause for a disease, and also understanding pathophysiology, the physiology of the pathogens that are responsible for causing the disease, understanding the pathophysiology. Through conventional way of diagnosis, through what we call it as serum and urine analysis, through serum and urine analysis. This is the earlier way. Through serum and urine analysis, you cannot go for early diagnosis. Early diagnosis is not possible. You know that most of the diseases that are caused in humans are by bacteria and viruses. So you know that most of the human diseases will be caused by this. See, these diseases caused by bacteria and viruses, we can be known only when symptoms appear. Only when symptom appears, then we go for urine and serum analysis, and then we can know the bacteria. It is not possible to diagnosis of this before the symptom. By the time symptom appears, there will be a large number of the bacteria and viruses in the body. Okay, what exactly we are right now discussing? We are discussing about a concept called as molecular diagnosis. So, diagnosis is a procedure to find out the cause for a disease. Under that, any treatment of a disease, proper treatment of a disease, require early diagnosis and understanding pathophysiology, means the physiology of that pathogen that is responsible. Through conventional conventional diagnosis, we go for serum and urine analysis, blood analysis or urine analysis we go for. In serum analysis and urine analysis can be done only when symptom appears. Then we will know which bacteria or which virus. But once the symptom appear, already their number will be much higher, much larger in the body of the patient. If we are able to find out the bacteria and viruses earlier to the appearance of the symptom, then it is easy for us to treat a disease. And then there are 3 techniques that are helpful in this regard. 3 techniques. Number 1 is recombinant DNA technology. Recombinant DNA technology. The second one is polymerase chain reaction, which we abbreviate it as PCR. The third one is ELISA, ELISA. As I expanded this earlier, this is nothing but enzyme linked immunosorbent assay, ASSAY. These are the 3 techniques. And so we are going through with molecular diagnosis of diseases. For a proper treatment of the disease, one should have early diagnosis, earlier, before the symptom appears. If you are able to diagnose the disease and understand the physiology of the pathogen, that is the one that is responsible for causing a disease. But earlier methods, like whatever the methods we are going in hospitals, like testing the blood and testing the urine, through this the pathogens like bacteria and viruses can be detected, but only when the symptoms of that particular disease appear. By the time the symptoms appear, their number is already much higher in the body. If by chance, if you can go for the diagnosis of the disease much earlier to the appearance of the symptom, and their number will be quite less, and it is easily controllable. Whether we have any system relating to the technique, relating to the technology of detecting the pathogen earlier to the symptoms appear in a patient, in such a case, we have 3 technologies like recombinant DNA technology, number 1, polymerase chain reaction, that is PCR, number 2, ELISA, enzyme linked immunosorbent assay, 3 techniques. And coming on to this point, how this polymerase chain reaction can detect You know that we have studied this earlier. These two concepts we have studied earlier chapter. With reference to polymerase chain reaction, you know that very small amount of DNA can be amplified into billions of copies. Okay, you know that there are 3 steps here in polymerase chain reaction, like denaturation, then priming, especially annealing, you can say, and then extinction. Denaturation, annealing and extinction, they are the steps where a single copy of DNA can be made into billions of copies. Our question will come, how the technology will be helpful for the molecular diagnosis, how it is helpful to detect that. And especially, already this PCR technique is helpful to find out presence of HIV in case of AIDS patients. AIDS patient. So presence of HIV positive can be detected by this PCR technique. In PCR technique is used to detect the HIV in AIDS patient. It is also helpful to know about a mutated gene. You know that the mutated gene will result in cancer patient. The third one is, this PCR is an important tool in the discovery of several genetic disorders. Let us elaborate on this. So, polymerase chain reaction, which will be normally used to amplify the DNA, can be used to detect the HIV in AIDS patient. It can also helpful to detect the mutation that has occurred in cancer cells. It is also helpful in discovery of several genetic disorders. See, you just imagine, let's elaborate. Elaborate on that. We have a single stranded DNA, or sometime it can also be RNA. This is tagged with radioactive molecule. This single stranded DNA is allowed to hybridize with the DNA present in clone of cells of a patient, suspected patient, clone of cells. What we'll have? We'll have a single stranded DNA, or it can be RNA, tagged with radioactive molecule. Then naturally this single stranded is called as a probe. You know that we'll have a known sequence that will be allowed to hybridize with the DNA of the clone cells of a patient. And when such hybridization takes place, after that it is autoradiographed. Autoradiographed. You can say autoradiography is nothing but exposing that hybridized DNA to X-ray film. You know that since the known sequence of DNA, you know that it will going to hybridize the known sequence of the DNA of the patient, of the humans, but you know that the mutated genes, mutated gene, or you can say the mutated gene that is part of DNA, which has undergone mutation, which is cause for a disease, cannot hybridize. It will not hybridize. Hybridize means it will not become double-stranded. It remains a single-stranded because it cannot be the complementary for this known DNA probe. And thereby it can be identified in that photographic film. In the photographic film, you can identify that mutated gene and thereby you can know about the disease. You can know about what exactly the mutation has undergone. This is what exactly the technique. I just repeat. I was telling you that PCR technique will be helpful in detecting the presence of HIV in AIDS patient. It is also helpful to detect the mutation that has occurred in cancer cells. And also it is a best tool to discover several genetic disorders. Procedure: a single-stranded DNA or RNA is tagged with a radioactive molecule, and naturally it will act as a probe. Its sequence is known sequence. This probe is allowed to hybridize with the DNA which is present in the clone of cells extracted from the patient. After hybridizing them, if it is autoradiographed, exposed to X-ray, the known sequence of DNA will hybridize with this, but the mutated genes that may present here will not hybridize. So thereby hybridized will be in the form of dark bands. The one not hybridized will not appear in the photographic film. So thereby it is helpful for us to detect that particular. This is the thing. And then with reference to this ELISA, enzyme linked immunosorbent assay, and basically you all know that ELISA, it is dependent on antigen and antibody, antigen and antibody reaction. We all know that the HIV that enters our body will naturally be an antigen. Antigen can be either a protein, that is a coat of this protein, or even a glycoprotein. glycoprotein. Naturally, it induces the formation of the antibody in our blood. See, presence of antigen, or otherwise the presence of an antibody will indicate of what is nothing but the HIV. So only when antigen is present, then only antibody is produced. If in the blood, if the antibodies are present, what is the indication naturally? Natural indication is the antigen is present. What a simple concept. ಎಲ್ಲೋ ಒಂದು ಕಡೆ ಹೊಗೆ ಬರ್ತಾ ಇದೆ ಅಂದರೆ ಅರ್ಥ ಏನು? ಸ್ವಲ್ಪ ಕೆಂಡ ಇದೆ ಅಥವಾ ಬೆಂಕಿ ಇದೆ. ಇಲ್ಲಿ ಸುಮ್ಮನೆ ಇಮ್ಯಾಜಿನ್ ಮಾಡ್ಕೊಳ್ಳಿ, ಬೆಂಕಿನ ಆಂಟಿಜನ್ ಅಂದ್ಕೊಳ್ಳಿ, ಆ ಹೊಗೆನ ಆಂಟಿಬಾಡಿ ಅಂದ್ಕೊಳ್ಳಿ. ದಟ್ಸ್ ವಾಟ್ ಎಕ್ಸಾಕ್ಟ್ಲಿ ದ ವೆರಿ ಸಿಂಪಲ್ ಕಾನ್ಸೆಪ್ಟ್. HIV ಇದೆ ಅಂತ ಟೆಸ್ಟ್ ಮಾಡೋದಕ್ಕೆ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಫಾರ್ ದಿಸ್ ಟೆಸ್ಟ್, ಎಂಜೈಮ್ ಲಿಂಕ್ಡ್ ಇಮ್ಯುನೋಸಾರ್ಬೆಂಟ್ ಎಸ್ಸೆ. ಕೇಳೋದಕ್ಕೆ ಭಯಂಕರ ಅನ್ನಿಸ್ತದೆ, ಆದರೆ ಏನು ಕಾನ್ಸೆಪ್ಟು? ಇದರಲ್ಲಿ ನಾವು ಡಿಸ್ಕವರ್ ಮಾಡ್ತಾ ಇರೋದು ಏನು? ಐದರ್ ವಿ ಕ್ಯಾನ್ ಡಿಸ್ಕವರ್ ದ ಆ್ಯಂಟಿಜನ್ ಸಿಗಲಿಲ್ಲ, ಅದ್ರ ಬದಲು ಆ್ಯಂಟಿಬಾಡಿ ಇದೆ ಅಂದರೆ ನ್ಯಾಚುರಲಿ ಆ್ಯಂಟಿಜನ್ ಹ್ಯಾಸ್ ಟು ಬಿ ಇದೆ. ದಟ್ ಈಸ್ ವಾಟ್ ಎಕ್ಸಾಕ್ಟ್ಲಿ ದ ಥಿಂಗ್. So this will help us to detect the pathogen earlier. And then this is what exactly the concept of molecular diagnosis that will replace the conventional diagnosis by serum and urine analysis. Whatever cannot be found by serum and urine analysis can be clearly detected using these techniques. That is the concept. Next concept. Next concept. We have a concept by name gene therapy. Concept by name what? Gene therapy. Therapy, you all know, it is nothing but treatment. Therapy andre eno? Treatment. Gene therapy andre eno? Treatment of a genetic disease. First of all, why there will be what is a genetic defect ಅಥವಾ genetic disorder ಹಂಗಂದ್ರೆ ಏನು? ಒಂದು ಜೀನ್ ಅಂದರೆ ಇಟ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ DNA. ಅದೇನು ಮಾಡುತ್ತೆ? mRNA ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ. How mRNA is produced from DNA? Through transcription. After that, what exactly the mRNA undergo? It will undergo translation. What is produced? Protein. What is the gene product? Protein. The genes are there to produce the proteins. Normally. Just imagine if this protein is not produced. Then what we say? There is a defect in gene. That defective gene, it is called as genetic defect or gene disorder, genetic disorder. You know, we have studied something like sickle cell anemia, that's how this genetic defect can be cured. See, this defect is due to gene which is not functioning. Gene is not functioning means what? gene is not producing, gene is not coding for a protein. So the gene is not functioning. What we have to cure this, what we have to do? We have to replace this faulty gene from a functional gene. ಏನು ಮಾಡಬೇಕು? ಈ ಜೀನ್ ಬದಲು, ಡಿಫೆಕ್ಟಿವ್ ಜೀನ್ ಬದಲು ಫಂಕ್ಷನಲ್ ಜೀನನ್ನು ಆ ಪರ್ಸನ್ನಿಗೆ ಇಂಟ್ರಡ್ಯೂಸ್. ಒಬ್ಬ ಪರ್ಸನ್ನಲ್ಲಿ ಏನಾಗಿದೆ? ಜೀನ್ ಡಿಫೆಕ್ಟ್ ಆಗಿದೆ, ಪ್ರೋಟೀನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಾ ಇಲ್ಲ, ಆಯಿತಾ? ಅದಕ್ಕೆ ಕಾರಣ ಜೀನ್ ನಾಟ್ ಫಂಕ್ಷನಿಂಗ್. ಈಗ ನಾವೇನು ಮಾಡಬೇಕು? ವಿ ಹ್ಯಾವ್ ಟು ಇಂಟ್ರಡ್ಯೂಸ್ ದಿಸ್ ಟು ದಿಸ್ ಪರ್ಸನ್. Then what will happen? So then naturally the gene will produce a protein. So what has done? The gene defect is cured. Are you following me? ಒಂದು ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ಗೆ ಕಾರಣ ಜೀನಿನ ಡಿಫೆಕ್ಟ್. ಆ ಡಿಫೆಕ್ಟಿವ್ ಜೀನ್ ಬದಲು normal functioning gene, if you are able to replace it, then you are curing the genetic disorder. That is what exactly required. Then how, in what method a gene can be transferred? So all those things. So with reference to all these things, okay, very first attempt for gene therapy was made in the year 1990. And it was on 4 years old girl. 4 years old girl. Extra information, it's not there in your textbook. Her name was Asanti De Silva. Asanti De Silva, extra information. And it was by Dr. treated by W. Anderson. Extra information. And that girl was suffering from ADA deficiency. Was suffering from ADA deficiency. The Asanti De Silva. Who is Asanti De Silva? A 4 years old girl. Okay, and she was suffering from ADA deficiency. What exactly meant by ADA? Adenosine deaminase. What is meant by ADA? Adenosine deaminase. What is the adenosine deaminase? It is nothing but an enzyme. Okay. And what is meant by ADA deficiency? This is not produced. In whom? In Asanti De Silva. Why? The gene for ADA synthesis became defective. That is why this enzyme is not produced. Okay. So, what the concept we are right now discussing? We are discussing a concept by gene therapy. What is meant by gene therapy? It is the treatment for a genetic disorder. First of all, what is a genetic disorder? A defect in gene in an individual. What the gene is responsible? Gene is nothing but DNA. It was producing a protein, and in that person genetic defect, that protein is not produced. Why that is not produced? Because the gene responsible for that is defect. Then how to correct it? Replace that defective gene from a normal functioning gene. What you are supposed to do to cure a gene therapy, a gene disorder? It has to be that normal functioning gene has to replace that faulty gene. Okay, that is what exactly the basic concept. Now what we are discussing? We are discussing the very first attempt of gene therapy. When this was the first gene therapy was done? It was done in the year 1990. And it was done on whom? It was done on a 4-year-old girl. And what was her name? Asanti De Silva. Who conducted this gene therapy? Dr. W. Anderson. It was done in USA. It was done in USA. What was that girl was suffering from? She was suffering from ADA deficiency. What is meant by ADA? ADA means adenosine deaminase. What is meant by adenosine deaminase? It is nothing but an enzyme. And in her, what was the problem? This enzyme was not present. Why this was not present? Because the gene coding for that in her became a defective and it was not producing. Okay. In all of us, whether ADA deaminase is present or not present, why this is required? This is required for the proper functioning of immune system. This particular adenosine deaminase is responsible for proper functioning of the immune system. And in our case, this immune system is not functioning. So then, what was the possible thing? What was the possible thing? See, the first possible thing is bone marrow transplantation. Possible thing, bone marrow transplantation. In gene therapy, one can go for bone marrow transplantation. The cells of the bone marrow, especially responsible for producing this, can be transplanted. Okay. Or, so what is the second concept? Enzyme replacement therapy. Enzyme replacement therapy. That is, the functional ADA, that is enzyme, is injected to patient. ಈ ಎರಡು ಟೆಕ್ನಿಕ್ಸ್, ಒಂದು ಬೋನ್ ಮ್ಯಾರೋ ಸೆಲ್ಸನ್ನು ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡಿ ಅವನ್ನು ಟ್ರಾನ್ಸ್ಪ್ಲಾಂಟ್ ಮಾಡು, ಎರಡನೇದು ಎಂಸೈಮನ್ನೇ ಡೈರೆಕ್ಟಾಗಿ ಇಂಜೆಕ್ಟ್ ಮಾಡು. ದಿಸ್ ವಿ ಕ್ಯಾನಾಟ್ ಹ್ಯಾವ್ ಕ್ಯೂರ್ ಪರ್ಮನೆಂಟ್, ಇದು ಟೆಂಪ್ರವರಿ. ದಿಸ್ ಕ್ಯಾನ್ ಬಿ ಟೆಂಪ್ರವರಿ. Okay. In this case, in Asante de Silva, what the doctors did was, they took out that lymphocytes from the bone marrow. Took out the lymphocytes from bone marrow, and they isolated from outside the body. They took out those lymphocytes of the patient. And then the functional ADA gene, it is nothing but DNA, ADA gene, with the help of retroviral method, reverse transcription method, and they introduce this into these B lymphocytes. The functional ADA genes were introduced into the lymphocytes which are taken out from the body of the patient, and then when these genes are incorporated, they cultured this, means they made multiple copies, and after that culturing, they injected this to the body of that 4-year-old girl. I just repeat. What was the gene? How was the gene therapy was done with that 4-year-old girl? They took out that lymphocytes from the bone marrow of that girl, and you know that their number was increased. To those lymphocytes, the functional ADA genes, that is, we call them as complementary DNA, CDN, because the complementary DNA. So these were incorporated into this, and then they are cultured, they are made into large numbers, and after that they were injected back to this girl. And since it was having the functional gene, it produced the enzyme, and thereby she got cured. But these lymphocytes are not immortal. ಇಮ್ಮಾರ್ಟಲ್ ಅಂದ್ರೆ ಏನು? ಶಾಶ್ವತವಾಗಿರೋದು. ನಾಟ್ ಇಮ್ಮಾರ್ಟಲ್ ಅಂದ್ರೆ ಏನು? ಬಹಳ ಕಡಿಮೆ ಅವಧಿವರೆಗೂ ಇರೋದು. Why these are not immortal? They have a short lifespan. They will be there for about 9 to 10 days. Their lifespan is lymphocytes, even the cultured lymphocyte can live for about 9 to 10 days. Afterwards, they will die. So what has to be done? Whatever the one which have been increased, cultured, they can be cultured more and more. Periodic infusion, time to time, this has to be infused to this girl until she leaves. This has to be the procedure. Then also, it is not a permanent cure. Of course, it is not a permanent cure. How one can attain a permanent cure? How one can go for a permanent cure in this regard? Just imagine, in the early embryonic stage, if the bone marrow cells that were producing this are incorporated into those cells, if this defect is known earlier at embryonic stage itself, when the number was very less, If these ADA producing bone marrow cells are incorporated or introduced into those embryo cells, all the embryo cells, then there can be a permanent cure, not otherwise. Okay. What's the concept we have discussed here? It is the first gene therapy attempt. Okay. So, let us once again rewind this. What the concept we are discussing? Gene therapy. So, in that gene therapy, what exactly we are discussing? We are discussing the first attempt of gene therapy. When was this first attempt of gene therapy was done? It was done in the year 1990. Who was the patient? 4 years old girl. What she is suffering from? She was suffering from ADA deficiency. What is the full form of ADA? Adenosine deaminase. What is meant by adenosine deaminase? Adenosine deaminase is nothing but an enzyme. Why this enzyme is required? This enzyme is required for the proper functioning of immune system. If this enzyme is not present in the body, what will happen? The immune system will not function properly. So this ADA deficiency can also have one more name. It is also referred as SCID. SCID. It is nothing but severe combined immunodeficiency. ADA deficiency can also be called as severe combined immunodeficiency. This name is not given in your textbook. Why there was ADA deficiency in that girl? Because the aDNA gene responsible for this enzyme production was defective. And the scientists, what exactly they did? They isolated lymphocyte cells from the bone marrow of that patient outside, where they cultured it. They also introduced functional ADA gene, that is nothing but complementary DNA, what we call that as, into those lymphocyte cells. And after that they cultured it. They made large number. And periodically they were given to that patient. Whether this resulted in the permanent cure to that girl? No. Why? Because the lymphocytes, what they are injecting to that girl, were not immortal. What is meant by immortal? Immortal means they live forever. Not immortal means they will live for a very short duration. What is the lifespan of these B lymphocyte? It may be 9 to 10 days, very short period. In such a scenario, what is the other alternative? The other alternative is those cultured cells periodically has to be infused to that particular curve. Whether there can be any permanent cure for this? Of course it is there. Then how? Then the bone marrow cells containing the functional ADA gene has to be introduced to the embryonic stage, all the cells of the embryonic stage, or you can say embryo. In such scenario, it can have a permanent cure, otherwise it cannot. So, under field of medicine, what are the things we discussed? Biotechnology application in the field of medicine. We discussed about recombinant therapeutics, and we discussed about insulin production. We discussed about molecular diagnosis, and we discussed about the gene therapy. And then in this chapter, we will move over to transgenic animals. In this particular chapter of biotechnology applications, now we are moving over to transgenic animals. See, just imagine a gene from other source is introduced into an animal. Then such gene is called as transgene. The animal to which the transgene is introduced is called as transgenic animals. What are the animals they have produced from with the help of transgenes? We have transgenic mice, transgenic rabbits, transgenic cows, transgenic rats, transgenic pigs, transgenic fish. Several of them are produced. 95% of transgenic animals that are produced are mice. You know that for experimental purpose, several transgenic mice have been produced. What is the requirement of this? Why should one have a transgenic animal? What is the purpose behind it? The purpose behind it is for several things. Let us just stop here at this class. Then we will continue from this particular concept in our next class. Thank you.